Thank you for the invitation to come in and speak before you. I love doing these sorts of things, the kind of public outreach of science, because I think so many of the things that are happening at the Broad are really exciting, really cutting edge, and really changing the way that we think about disease, about treatment, about therapies, about classification. It really cuts across a huge range of disciplines, and I think this series does an excellent job of showcasing just that. Now, I myself am a statistical geneticist by training. I've worked in psychiatry and psychology for the entirety of my career as a statistical geneticist over the course of the last, say, 12 years or so, something like that. And I'm going to talk to you about the progress that we're making in psychiatric genetics, because this, like so many other areas of disease, is really being revolutionized by the technological developments that are occurring around interrogating the human genome. And what we're learning is in some ways surprising, and in some ways is kind of answering, I think, in some instances, a century-old debate about the structure and nature of disease in the population. So to start, I'd like to just give you a little bit of a brief history of schizophrenia, okay? And really, when we talk about schizophrenia, the initial articulation of a classification system of madness, as it was originally described, occurred in the kind of late 19th century, early 20th century, by a guy named Emil Kraepelin. So he delineated between two forms of madness, this idea of dementia praecox, or precocious dementia. So this is kind of like an early onset cognitive decline. And then the second is this manic depressive illness. And those terms got shifted over to what we call now schizophrenia and bipolar disorder. And this was a clear dichotomy, a delineation in the classification of mental illness as we understood it at that time. When you initiate a field and you kind of split out two disorders, you get to kind of lay claim to some of the nomenclature and some of the structure. That's a natural sort of thing. But one of the primary arguments that was used by Kraepelin about why these two diseases really were separate diseases came around heredity and familiality, this idea that there was a breeding true of schizophrenia or bipolar or dementia praecox or manic depressive illness within different pedigrees. So if you were a case of schizophrenia, it seemed much more likely that you came from a family that has schizophrenia rather than the reverse. And in addition to this kind of observation of breeding true, there was a kind of implicit that moved to a slightly more explicit statement that these diseases, that both schizophrenia and bipolar, are actually based on biological causes, right? So this isn't something about your parents not treating you very well or something about your environment, that there is some biological basis to this. And by invoking the argument about running in families, this argument then suggested at some level a genetic link. Well, a little bit after that, Eugene Bleuler said, well, wait, not so fast. We got to kind of hold the bus, if you will, although he probably didn't say that a century ago. Dementia praecox is a kind of lousy name, because if we take these patients that kind of tick the boxes for schizophrenia, they have a kind of lose touch with reality, they have delusions or hallucinations, they have these different clinical features that we describe and document, it's not a guarantee that we're going to have this cognitive decline over time. So this really, dementia praecox, was based on this argument that if you followed schizophrenics over the course of their illness, you saw this reduction in cognitive abilities. And while that's certainly true on average, it is not de facto the case that people that present with schizophrenia will lose their cognitive abilities. And so that's a kind of bad name. So maybe we should call it split-brained or schizophrenia rather than calling it dementia praecox, which I think people generally agreed was a little more sensible. But he also articulated 4 A's for schizophrenia. Doctors like these kinds of mnemonic devices because you have to remember a huge body of knowledge. And so these 4 A's of abnormal associations, autistic behavior, we'll come back to that, but the idea that you're going to withdraw into yourself, so this is what we call negative symptoms now with respect to schizophrenia. Abnormal affect, so the way you interact with people, your kind of general demeanor is a bit strange. It doesn't fit with what we would think of as normal variability in the population. And a certain amount of ambivalence, like just I don't care, it doesn't really matter, what's the whole point of it altogether. Those were the kind of clinical features that Bleuler really tried to put pen to paper into kind of refining the definition of schizophrenia at that time. again, there was this general agreement about the biological basis for this disease. Now, the other thing that Bleuler said was that basically it's not one schizophrenia, it's a group of schizophrenias. So he clearly articulated this idea that there were lots and lots of little different kinds of diseases running around the population, masquerading as this global construct of schizophrenia more generally. Now, when we talk about any complex trait, not just behavioral and psychiatric traits, it's worthy to really take a moment and talk about Francis Galton. So Francis Galton is an amazing individual, kind of classic Victorian gentleman scientist, definitely fits into this like polymath, arguably Renaissance man. He invented weather maps, like those isobars and all that kind of stuff. That was a kind of Galtonian innovation. But probably one of his most important and hallmark contributions was the development of something called regression. And regression, which you may or may not have heard of, is like the statistical analytic technique that most of us use, at least at a starting point when trying to understand the relationship between two variables in the population. But he developed regression to try and answer this question about the idea of there being heritability, there being this genetic variability in the population, and that genetic variability that is in the population translates to phenotypic variability that we observe. So there's something about the variation in our genes that causes the variation in the traits that we look at. And this was a very powerful idea, and was certainly building off of a lot of work that his cousin, someone else you might have heard of, Charles Darwin, kind of propounded in the kind of theory of evolution. So this is a nice sister theory in some ways to try and get at that. More formally, there's this idea of the twin method. So the twin method has a very, very deceptively simple logic. It is that identical twins are identical genetically, and fraternal or non-identical twins share about half of their genes on average. And by comparing those two different types of twins, you can estimate how much the genetics is really contributing to your phenotype, okay? And this was a really, really important contribution because it gave us a way epidemiologically to look at different kinds of twins and then ask questions about how heritable is a disorder based on how similar identical twins are in contrast to non-identical twins. The earliest studies for schizophrenia started in around 1941 for twin studies. There have been thousands upon thousands of twins studied along these lines, and they probably reached their kind of pinnacle of importance and critical mass in sometime in the 80s to maybe early 90s, something like that. And you can see a couple of these different papers down there for more information. Then 25 years in the wilderness. So we get to like the 90s, we're like, well, we're genetic, we've done these big twin studies, we're really convinced that there's a genetic contribution to risk for schizophrenia in the population. Can we find it? So we start with this idea called segregation analysis. What segregation analysis tried to do was deconstruct the pattern of inheritance in families and use that as a way to figure out whether it was like a Mendelian disease like cystic fibrosis where it's recessive, or whether it was a Mendelian disease like Huntington's where it was dominant, or somewhere in between, and that failed. We did linkage analysis where we kind of basically look at the pattern of association of phenotype to specific positions in the genome across large pedigrees, and that failed. We looked at candidate genes. We're like, well, we know lots about neurobiology, I promise. I think we got it sussed out. These are genes that we should interrogate. We should try and understand what genetic variation in those genes contributes to disease, and that failed. So you might be getting this kind of theme that this 25 years in the wilderness was characterized by a lot of failure, unfortunately. But necessary, because if you don't try, you don't know, I think is a good way to think about it. But this still doesn't relieve us from the fundamental pressing need with respect to schizophrenia, that the drugs that we use to treat schizophrenia right now are on the order of about 50 to 60 years old. We don't have any novel mechanisms of therapeutics at all. There's no real progress being made in understanding how we can treat these patients more effectively. And our understanding of the basic biology is still pretty limited. We have some ideas. We know that the antipsychotics that we have target DRD2, but that's kind of about it. We have some hypotheses about circuits in the brain and different biochemicals and all those sorts of things. But we don't really grasp what is happening with respect to the biological basis of the disease itself. So like many areas of science, this is one of those technology to the rescue sort of things. And what we've seen transpire over the course of the last, say, 15 years is a massive revolution in human genetics, from the mapping of the human genome in 2001 to moving towards the International HapMap project from 2003 to 2007. And what the HapMap project did, so the human genome was like, here's the map of the genome, here's your reference, now we can start to, put things on top of that. Well, HapMap said, there's something else that we might be a little bit interested in rather than simply the sequence of the human genome, and that's the pattern of variation that we observe. So what are genetic variants that we see? I'll explain in a little bit what I really mean by each of these different kinds of genetic variants that we might be looking at. But what the HapMap did was it created this big bolus of genetic variation that is common in the population. Okay, so most everyone has a reasonably large fraction of variable sites that are shared with their neighbors, almost regardless of what population that they're from. And then this gave rise, at the same sort of time technologically, to a technique called genome-wide association. And now what genome-wide association is doing is it's interrogating these common variants, these single nucleotide polymorphisms, these single base changes in the human genome, and asking whether or not they associate with disease. Okay, so because this is a little bit of a kind of more teaching venue, and we're trying to learn and think about genetics and genomics and biology and all of the exciting things that's happening, just to refresh your memory, you have your cells, and then in your nucleus you have your chromosomes, and then the chromosomes have genes in some regions and not so much in other regions, but we're talking about 3.3 billion bases, give or take, something like 18 to 20,000 different genes, depending on how you want to do the accounting. And then there are maybe 10 million plus common variable sites. And maybe it's more, maybe it's less. You can argue about 30 or 10, depending on where you want to draw the frequency line. But basically what we're talking about is a huge amount of common genetic variation that is shared across most continental populations. So if you ask, is this site a variable site in Africa? A lot of the times it will be a variable site in Europe or in Asia or in South America or wherever you happen to go and investigate these genetic variants themselves. So just a few more definitions, a little bit more teaching. So we can think about having two chromosomes. So you get, the half of your DNA that you inherit from your mom or your maternal chromosomes, and then you get the half of your DNA that you inherit from your dad or your paternal chromosomes. And we have, here's our little gray reference sequence, and then we've got some variable sites here in red. Okay, so these are single nucleotide polymorphisms, single base changes that are variable in the population. So when we talk about a SNP, that's really exactly what we mean. These places where bases can differ between different individuals. Now, some people might have the TT genotype. So when we say genotype, what we mean are the two alleles at the position. Some people might be heterozygous or have the two different alleles, this TC, so this is a TC polymorphism, while other individuals might be CC, so they might be the other form of homozygote, so the two of the same alleles at that position. We can also think about those specific alleles, so each of those individual bases that you have at these variable sites in the genome are your alleles. And then you can also think about your haplotypes. So if we just extract the genetic variation from these chromosomes, we can slot them together so that you have this TAA haplotype and this CGC haplotype, okay? And so these are the variable sites that we're looking at in the population. And these are what we're testing when we do the genome-wide association kinds of studies. Now, when we do an association study, what do we mean by that? Well, it's a pretty simple design at its most, kind of basic. You can do this with a number of different statistical techniques, but the name of the game is basically you get a pile of cases, you get a pile of controls, and then you ask about the differences in the frequencies between those alleles in those two groups. And the thinking goes, genetic variants that contribute to variability in risk to the disease should be more common in our case pile rather than our control pile. So it's just a straightforward a test of differences in the mean frequency between these two groups. And that's our just general association test. And it's very simple to do once. It's maybe a little bit more difficult to do 10 million times, but now a relatively straightforward activity. So lather, rinse, repeat, do association, lather, rinse, repeat, do association, do that 10 million times. We have to use computers. It would take, I think, a lot of handwriting hours to do these tests by hand. And so what we get from these kinds of association scans are basically plots of minus log 10 p-value. So when we say minus log 10 p-value, what we really mean is just the strength of evidence for association. So the larger your minus log 10 p-value is, the smaller your p-value is, the stronger your evidence for association, the less consistent with the null hypothesis that there is no relationship between this SNP and risks for disease in the population. So we plot these things because we like to look at data, and visualization is good. So this was from the ISC group, the International Schizophrenia Consortium in 2009. And this red line is our kind of magic line of significance. For us to be reasonably confident that a SNP is actually relevant, it's got to have a p-value less than, kind of above that kind of red line. So more significant than that red line. And at, 2,600 cases and 3,300 controls, which at the time represented the largest single genetic study ever conducted for schizophrenia, there was no association. So maybe this isn't going to work. Maybe this is a bit of a problem. Not so fast, common variation people. This isn't a guarantee. Well, if we can't hang together, we'll hang separately, I guess, is one way of thinking about this. Another, perhaps more positive way of thinking about it is that there was a large-scale collaborative effort initiated by that man right there, Patrick Sullivan, to try and bring and aggregate all of the world's data of genome-wide association together under the same, basically, analytic roof. Okay, so what we're going to try and do is we're going to try and bring all of these disparate groups together and convince them to work together, convince them that sharing data is really important. This slide here convinced people that it wasn't going to be the case that you run, 2,000 or 3,000 cases and 2,000 or 3,000 controls, and you find a bunch of new associations that you can then go and try and chase down the biology. Instead, it's going to require us all working together, all coming together as a community and sharing our data. And this was a real shift. Like in the linkage days, you could get 3 or 4 pedigrees, map a disease, and then go work on the biology and write your nice scientific paper. And so there was a kind of general movement towards slightly more siloed or isolated work. And this is a very different model for scientists. This is more like how physicists interact rather than molecular biologists historically interact. So the current chair of the PGC schizophrenia group is Mick O'Donovan, and this is Stefan Ripke, who works in our group, and Mark Daly is the kind of analytic senior master of puppets, if you will. Now the PGC has 300 plus investigators, 80 different institutes, 20 different countries. There's been this is a pretty big, complicated operation, even by normal bureaucratic standards, and it really wouldn't be possible without the serious contribution from NIMH and really the kind of work done by Thomas Lehner and Pat Bender in convincing all of these different people to work together definitely helped. Okay, so PGC1, the past, as it were, 3 years ago now. Things move quick in genetics. Remember that. It's like an exciting time. We have now 9,000 cases, 12,000 controls, a handful of genome-wide significant sites. So the first kind of rays of sunshine coming down, manna from heaven, these small SNP effects that are emerging, still quite exciting, but not really seal the deal. You could be a skeptical scientist and say, maybe these things aren't actually real. Maybe it's just some bias or confounding that you haven't taken account of, that you don't really understand. And so, we'll just kind of persist. And this was the unveil at the World Congress of Psychiatric Genetics in 2012. And the World Congress is the big psychiatric genetics meeting. It's where most of the people interested in psychiatric genetics as a kind of profession. It is their professional meeting to come together and share data. And during the PGC meeting, when this slide was shown, there was a spontaneous round of applause that burst out in the room. Like, it's the first time that I've seen scientists almost overwhelmed by data. Like, there was just so much enthusiasm and excitement because this was the slide that convinced us that maybe we've actually sealed the deal in proving that common variation really does contribute to your risk to schizophrenia in the population. And that's a really important and powerful concept. We continue these activities, and now we're at 97 plus genome-wide significant loci. You get to about 108 if you include replication. And each of these things are single nucleotide polymorphisms, single base pair changes in almost all instances, scattered across the genome, that there's no way that we would have chosen to look at them a priori. These are things that are new, that we are learning, that is teaching us new things about biology, that is teaching us new things about how to think about the risk for disease in the population. So here's it in the kind of stock portfolio sort of chart, if you want, low rate of return, low rate of return, and then, this is the kind of future when you are being sold a stock portfolio, but I think it's reality when we think about psychiatric genetics and gene discovery, and these variants are hugely informative. So just take for a moment to ponder the idea that a phenotype that we define simply by asking people questions. Okay, so there's a structured clinical interview. We ask them questions. We sometimes ask family members questions, or loved ones, or friends or colleagues questions about an individual's behavior, how they act. Okay, that's all we're doing. And we have achieved what I think some definitely thought was the effectively impossible, that there are common genetic variants that give rise to risk to that kind of phenotype. And that's a really powerful idea, not only in terms of trying to understand the biology of disease, which is really, really important because we aren't very good at that right now, but also in terms of changing the view of mental illness as a medical phenotype. Here we have clear genetic risk factors that we can point to that confer risk to this disease that we have documented, and we spend a huge amount of money caring for, something on the order of maybe 60 billion a year at current estimate in terms of lost productivity and patient care associated with schizophrenia. So that's just schizophrenia, and this is a really radical, in some ways revolutionary idea. It's very exciting. Common genetic variants can be found for these kinds of diseases. Just an important thing to think about. So, schizophrenia has identifiable common genetic variants that confer risk. It's really, really a change in the way that we think about this disease. This was a long time coming. It took a lot of work, thousands of man-hours, if you think about all of the patient collections and the generating of the data and the analysis of the data and all of those sorts of things. Over 100 robustly associated regions at this point in time, and that means that there are lots and lots and lots of effects, right? So these are very small perturbations on risk. They change your risk to disease a very minuscule amount, but it is there, it is detectable, it is robustly and reliably identifiable, and that's really important because that means that we can start to do lots of other fancy things with some of these things, like chasing the biology or seeing whether or not they confer risk to other phenotypes in the population. But the other thing to bear in mind is that everyone carries risk alleles. These are common variants. Some variants have frequencies of 40, 50%. I guarantee you that every single person in this room has at least one schizophrenia risk allele from this set of 100. It's a basic mathematical impossibility for you to have 0 schizophrenia risk alleles at this point in time. You might have a little more or a little less risk, and that's certainly, definitely the case, but it does mean that we all carry these alleles in the population. So, some encouraging signs to convince us that we're not just, squinting at mirrors or looking at smoke or something like that. One is that the region of the genome that is, the gene itself, DRD2, that's the target of antipsychotics, as I mentioned earlier, that's in a region of association. This is a promising sign, something that we find encouraging in terms of there being potentially other therapeutic targets that live in and amongst those loci that we have yet to appreciate. So there's the possibility that therapies could emerge from this set of work. The second thing that we like is that there's a role of the synapse based on pathway analysis. Well, the synapse is kind of important in the brain. There's a lot of things that synapses do in the brain, and so that gives us a little more confidence that maybe this is, some legitimacy with respect to unraveling those biological components. And then thirdly, there is a suggestion of a role of the immune system. Now, with respect to schizophrenia, there has long been a hypothesis about the role of the immune system in risk for disease, and current analyses are somewhat suggestive that there may be some merit for this particular hypothesis. it is nearly impossible to prove that because we can't really go and biopsy the brain or look at the brain's immune system. We're not allowed to do that, nor should we be. I mean, it's not something that's generally considered available tissue in the scheme of things. We're not drawing blood, as it were. So, having these genetic results start to shed new light into some of the possible biological hypotheses that are, out and amongst and walking around in the population of scientists working on these diseases. So what else can we learn from these different kinds of data sets, right? So we now have these common variants, we have some risk, we got lots of genetic variability in the population. Well, one of the other things is, we talked about twin studies and how we established heritability and kind of there being a genetic component. Well, we can do this now from looking at the genetic variation. We don't have to do this observationally via looking at identical and non-identical twins. We can actually say, are the common variants that we observe in a case of schizophrenia more similar to the common variants that we observe in a different case of schizophrenia? Yes or no? And that's just the general basic principle of trying to get an estimate of what we call heritability, is trying to understand how much variability in the population can be explained from genetic factors. And we can do this directly now. We can look directly at common variation. So there's schizophrenia, but there are some other mental illnesses out there, and we kind of want to ask questions not only about whether schizophrenia has common variation conferring risk to these diseases, but other phenotypes that maybe aren't at the position of having somewhere in the neighborhood of 37,000 cases collected and genotyped on genome-wide association arrays. Can we resolve whether there are genetic risk factors, or is it largely environmental, or is it a social construct, or what of all of the kind of reasons under the sun that anyone can imagine about where these psychiatric illnesses come from in the population? Can we start to gain some insight into that? And using that basic principle of the more similar genotype is between a pair of individuals, the more similar their phenotype or their disease status or their trait status should be, is a very powerful one that Peter Vischer and Naomi Ray and many others in Australia, along with a bunch of other folks throughout the community, really developed and articulated and put into these computational packages to start to ask those kinds of questions. And so to go back to that PGC idea, so the PGC doesn't work just on schizophrenia. It works on schizophrenia, autism, ADHD, major depression, bipolar disorder, as well as now a whole bunch of additional diseases that are going to come in the door, like anorexia nervosa, PTSD, obsessive compulsive disorder, Tourette's, all of these different mental illnesses are seeing what is happening in schizophrenia and wanting to capitalize on this same toolkit, because we really don't understand very much about the biology or how the risk for these diseases operates in the population. So here we're going to look at something like, an earlier freeze of the schizophrenia data, somewhere on the order of 33,000 cases of one mental illness or other, and 40,000 controls. And we're going to ask this first question at a univariate level, so on each phenotype at a time, whether cases of one phenotype are more similar than others. And so this is some work that Hong Li and Naomi Ray down in Australia have really been spearheading as part of the PGC, and it was recently published as the kind of genetic relationship between 5 psychiatric disorders, and I think you can guess where I'm going. But let's just start with the univariate case, the one phenotype case. And so here we have schizophrenia, bipolar, major depression, autism spectrum disorders, and ADHD. And for all of these phenotypes, for each one of these different diseases, we identify that common variation is a significant contributor to risk in the population. So that story that I told you about the identification of common variants for schizophrenia should well work across all of these mental illnesses, not simply schizophrenia. And that's really important, because this means that we can start to understand some of the biological bits and pieces that are relevant to these different diseases. So common variation accounts for some, not all, heritability. Reasonably standard amounts in the kind of order of 20 to 30%, much lower than the twin estimates. Why that's the case, we still don't know. It's kind of one of the open questions in the community. Why do we see this disconnect between family studies and the kind of measured genotype studies? And there are lots of possible reasons for that. But perhaps more of interest with respect to settling this kind of century-old debate of, was Kraepelin right? Was it correct to really distinguish between schizophrenia and bipolar? Are these two different diseases that will really breed true in the population? Is there any evidence for overlap or sharing between these two diseases? And so we can ask this same question, but we can do it in a bivariate case. And so we have the significant pairwise combinations ordered from, kind of left to right. So here are the significant ones delimited by this bar here. And we see that schizophrenia and bipolar share a huge amount of their genetic risk. That kind of legend reads around 0.7. So that's, maybe half of the variability in the population that causes schizophrenia that we capture for common variants also confers risk to bipolar disorder. So this Kraepelinian dichotomy between these two phenotypes wasn't perhaps that, realistic or accurate with respect to what is going on with respect to the genetic risk factors, at least as best as we can estimate at this point in time. It is also worth highlighting that the sharing does not just stop there. There is sharing between schizophrenia and major depression. There is sharing between bipolar disorder and major depression, between major depression and ADHD, between schizophrenia and autism. Lots of different pairwise combinations of overlap at the genetic risk level as measured from these phenotypes that we collect from clinicians, diagnosing as best they can across the world. And so this really underscores this idea that there may be a lot of shared genetic risk factors that are kind of central to risk disease, and then there may be some specific things that give you, say, schizophrenia or bipolar if you have some kind of overall liability or risk to these diseases at a high level. So specific effects do also exist. So we have this idea that there is some specificity. So if I try and predict your schizophrenia status and I use bipolar, I'll do a less good job as I will if I use just the bipolar analysis and the bipolar data that we use. And so there is a greater degree of sharing within the set of cases that are bipolar, that we call bipolar, the set of cases that we call schizophrenia. But there is also this idea, this kind of lingering sense that there is a bunch of shared different genetic effects more broadly. And that's another important idea because we don't, the classification systems that we have right now, they're built around, kind of a century of clinical practice with very little in the way of biomarkers or robust biological measurements. It's not like I can draw your blood and say, well, it looks like you got schizophrenia. That's not how this works. It's, I have to ask you questions. I have to put you in for observation. You have to talk to me. I have to talk to you. Or if you refuse to talk to me, I'll probably write that down too if I were a clinician. All of these different things kind of are the way that we define our diseases. And that doesn't necessarily reflect exactly how the biological basis of these diseases emerges from, in part, the genetics. Okay, so that's schizophrenia and other common complex disease. Now let's just take a moment to have a brief history of autism. So, another old white psychiatrist born somewhere in Central Europe. This one moved to America, so he's called American in Wikipedia, but he was born in what was Austro-Hungary and now I think is Ukraine or something like that. So Leah Connor. And he borrowed that term autism from Bleuler. So, psychiatrists like to appropriate terms from other psychiatrists and then declare new disorders. So they get the credit for defining the phenotype, but they don't have to come up with a new word. It's like the best possible world for creating a new phenotype. But the thing about autism is that there were a couple of very important differences. One is that this was something that was apparently there at birth, or at least very early on in life. So according to Connor, this was, basically you're born afflicted with autism. That's not completely true, as we've kind of appreciated over time, but it is certainly what we would consider an early developmental disorder. And the kind of phenotypic hallmarks of autism really kind of is a focus on self, a withdrawn from the population, withdrawn from social interaction, reduced social communication skills, and then these kind of repetitive stereotype behaviors, including things like hand flapping or can stacking or any number of different kind of stereopathies. So things that people do repetitively that are kind of unusual and not typical more generally. And so that's how we, again, clinically defined autism as a community. But that story has changed over time. So here's a big data slide, and it's kind of a bit scary. And this is looking at what the autism spectrum is and how it has evolved over time. So we have different years, okay? So we've got from, say, 1960 to 89. And then you can ask the question about what's the prevalence of autism spectrum disorders. So this is, like 3 out of 10,000, or maybe 4 out of, or 5 out of 10,000. So pretty rare. And then you can ask what fraction of the cases at that time had intellectual disability. And in the kind of 60s to late 80s, the overwhelming majority, 73.5% of cases diagnosed with autism, also had intellectual disability. If we move forward in time to, say, a slightly more modern view, 2005 or so, we now move from, rather than this kind of 4 or 5 in 10,000 to maybe 100 in 10,000, or maybe 1 in 100, or 1 in 88, or 1 in whatever it is now, 68, I think, from the observational studies from the CDC. And if you look at the fraction of patients with intellectual disability, that's dropped dramatically, okay? So what we've seen is a kind of widening of the tent of autism with respect to how we define it as a community of clinicians and scientists and all of these different things. And this kind of reflects some of the evolving thoughts about how we think about autism and Asperger's and the spectrum of autism disorders in the population. So once more into kind of the Galtonian twin and family study model. Here we see high sibling recurrence risk for autism. That's very consistent. We see parents and individuals showing higher endorsement of what we would think of traits consistent with autism. So they might not meet full diagnostic criteria, but they might be slightly less socially communicative than some of the, a random person drawn from the population. Or they might be slightly more inclined to engage in certain repetitive behaviors or what have you. So those individual low-level trait endorsements are higher in patient when you select based on patients. Again, this idea of genetic component to risk. And there's a familial aggregation of autism with other psychiatric disorders. So some of those cross-phenotype things that we were seeing earlier on with respect to, say, autism and schizophrenia, for example, that may also, be borne out by what we're observing epidemiologically, observationally. Okay. Now, there are a couple of things to think about when we think about genetics, and one of and in particular, genetic risk. And one of the things that's very important is what we call selective pressure. Okay, so evolution, if you do not have any children, okay, if you do not have any children, all of the genetic variation that's special to you gets lost from the population, right? So it's working out. So if you have fewer children, if you have this reduction in fertility or fecundity, that means that the alleles that you have are at a selective disadvantage. Evolution is not interested in keeping them around. And so this paper by Rob Power and colleagues from just last year took a look at, in Sweden, what the overall rate of children is compared to unaffected siblings of patients with different major psychiatric illnesses, ranging from schizophrenia and autism all the way through to depression and substance abuse. And what we see is that you have fewer children if you are the sibling that has the psychiatric illness compared to the sibling that does not have the psychiatric illness. And so that means that very big effect variants, so things that are Mendelian or near Mendelian or basically guarantee that you have a disease, they won't hang around the population very much because they're being selected against at a population level quite aggressively. So that means that strong effects are kept rare or are potentially even new. So they are de novo, newly arising mutations that your parents don't have, but you yourself do have. On the kind of flip side, effects that are very weak, they can be common, right? So things that have very, very, very minuscule effect on your overall probability of having children, they're perfectly okay to kind of float to much higher levels. And so when we were looking at common variation, we were looking at weak effects that are perhaps slightly more globally shared, but they exert, very, very little risk to disease in any individual or in the population in general. So autism, like schizophrenia, like insert your psychiatric illness of interest here, spent 25 years in the wilderness where segregation analysis failed, where linkage analysis failed, where candidate gene association analysis basically failed. And autism, in contrast to schizophrenia, is in an even worse position. There are currently no, fully effective or approved therapeutics for autism at this point in time. There's very little progress in understanding the biology, bits and pieces here and there, but over the kind of course of this 25 years, pretty limited insight. There have been appreciations of certain syndromes that people also say present with autism and some other syndromes. So there's something called Rett syndrome that you can go and look up if you're interested about trying to explore one example of a syndrome that presents with both autism and a whole bunch of other phenotypic components, other traits in the population. Okay, so once again, technology to the rescue, and what we're talking about now is doing next generation sequencing. So here our goal is no longer simply documenting all of the common variation that exists across the population. We want to capture nearly all of the genetic variation within an individual. And kind of one of the papers that's a really good example of how next generation sequencing can teach you a lot about a lot of different things is the 1,000 Genomes Project that, like with the human genome and many of these other, and the HapMap and other large-scale collaborative activities, the Broad played a major role in making this happen and helping to work with a whole international collaboration of scientists, again, coming together to understand things about genetics and human variation in the population. So here we sequenced 1,000 individuals over the course of about 5 years, and it was around $5,000 per individual, so a lot cheaper than the hundreds of millions of dollars that it cost to get the first human genome, albeit quite a bit later, so it's maybe not as exciting. But we're looking at 3 billion bases, give or take, and again, these genetic variants in the population are contributing to making us different. They're explaining some of the phenotypic variability, those traits that we look at like height or diabetes or schizophrenia or autism or what have you. Some of the variability in those phenotypes is definitely a consequence of the genetic variation that each one of us possesses. So again, we're trying to capture all of the variation, okay? And it's all in italics because it's not fully comprehensive. There's a whole bunch of weird and wonderful stuff in the genome. The genome is a very complicated place, just like biology is a very complicated place. It's just kind of the nature of the beast. But this captures nearly all of the genetic variation that might be conferring risk to diseases in the population. And when you're working on a complex trait like autism or schizophrenia, you don't have to capture everything ceaselessly because you know that as long as you capture most of it, you're very likely to at least capture some of the things that are going to confer risk. So we don't have to quite be so completionist as maybe some people who are chasing a single gene disorder or a single genetic variant disorder have to be, because if they don't find their variant, that means they won't find their disease-causing allele, and that's a different challenge at some level. Okay. So now we're going to talk about a slightly different paradigm. So if you remember the kind of reduction in fertility and fecundity, fewer children if you have a major psychiatric illness than if you don't compared to your sibling. What we're going to do is we're going to look for newly arising mutations. So these are novel variants, okay? So they're just going to pop up out of nowhere. And here we have our dad and our mom, and then we have this offspring here that might be a boy or a girl, and this is kind of the way we represent those things as geneticists. And they have A/A, so they're homozygous for that A allele, both parents, and then the kid has this C allele, okay? So this is a new mutation, something that's different that we haven't seen yet before in most instances. Not all instances, there's recurrent mutation and those sorts of things. And this is drawn from a bunch of different papers that have been published over the course of the last couple of years, trying to establish and identify novel genes for autism in the population. And what I'm going to be presenting is really based on the Autism Sequencing Consortium work from NIMH and NHGRI, as well as the Simons family, or the Simons Foundation, and a few other different sources of both data and support. Okay, so let's just talk about a gene just to refresh ourselves for a moment. So, genes are made up of exons. Those are our little bits and pieces. And then genes are transcribed, so they are copied into RNA. And then that RNA is translated into protein. So here's our DNA. We got these exons in this kind of gold color, and then our introns in this kind of green color over here. Picture I took from the internet somewhere, but fair use for educational purposes, so we're all happy about that. And the exons themselves are these kind of these 3 base pair codes. So when we talk about like the genetic code, Some of us really are referring to this idea of genes being turned into RNA being turned into protein. And the mechanism by which that is occurring is these exons have these 3 base pair motifs, and those are called codons, okay? So those codons encode for a specific amino acid, and those amino acids are then turned into proteins, okay? So this is our like central dogma of biology, really, really important, basic stuff, hugely important. And so when we think about coding mutations, it's important to think about the amino acid table, okay? So here it's a very complicated table, but it's not too bad. So we've got our first base of the codon here, the second base of the codon here, and then the third base of the codon over here, okay? So 4 letters, 4 choose 3, 64, et cetera, et cetera, et cetera. And so if we have TTT as our codon, that makes a phenylalanine, okay? So that's a specific kind of amino acid. And when we're looking at coding mutations, what we're looking at are single, in some instances, most instances, single base pair changes of a specific base in a codon. So if we have this T to C here, this TTT to TTC, that's the change in the codon, that we call a synonymous mutation. And we call that a synonymous mutation because if we look up in the amino acid table, we have exactly the same amino acid from this, prior to and posterior to that mutation. So whether it be a T or a C in that third base position in that codon, we get exactly the same amino acid. So we think most of those are pretty neutral, and evolutionary geneticists and population geneticists say, yeah, you're right, those are pretty neutral by and large, not always, but pretty much. However, we can also have a different amino acid. So if we have a base change here, we go from ATA to ATG. So if we change those different bases, we're now moving from an isoleucine to a methionine. Okay, so we've changed the amino acid. So we've now changed the sequence of amino acids, and that's going to have a change on the protein itself. Some of these are relatively benign. Some of them are very deleterious. It depends. We try and predict. We don't do a brilliant job yet of predicting what of these missense mutations are really relevant to changing a protein and which aren't. It's a very hard problem. It's a big search space, et cetera, et cetera, et cetera. But nevertheless, this is a likely more important class of mutations than synonymous mutations. Third, we also have this stop signal in our genes, right? So a gene's got to tell, tell it where the end is. We got to say, hey, we don't need any more amino acids. That's our protein. It's just the right length of protein, like the trees in Michigan or something like that, if you remember the presidential election. So here we go from this TAC to TAA. So that's another single base pair change. And we're moving from a tyrosine to a stop codon now. And so that's causing a premature termination of the protein, and that's expected to be much worse. So we're losing the function of one of the 2 copies of that gene in the individual, because they now have a stop codon there rather than the kind of typical amino acid or even a different amino acid at that position. So we can ask the question, classifying based on mutation type, whether we're seeing a higher rate of specific kinds of de novo mutants or not. And this is work that Caitlin Samoha and others in the group have really been spearheading. And here we have 3 classes of mutation. We got synonymous, missense, and loss of function. So those are the stop codons. They also include frame shifts. So these are when you have bases inserted or deleted. But it's another class, another kind of this kind of stop codon sort of acting mutation that we call a putative loss of function. And we see a significant enrichment in autism cases in blue compared to controls. So this is 500 odd controls that have been sequenced for just their coding region of the human genome. Compared to the expectation. So the controls look a lot like expectation for missense and loss of function mutations, and the cases have a clear significant enrichment. So we're seeing more newly arising missense mutations and more newly arising loss of function mutations in autism cases, in contrast to what we expect from modeling the mutation rate or what we expect based on looking at controls. this now gives us actual genes. This is a really kind of exciting change. You know, we have actual genes now being emerging as candidates, where we're seeing a piling up of these loss of function mutations. We're seeing a piling up of these missense mutations, much more than we would expect to see by chance. P-values on the order of 10 to the minus 10, which is well significant, even correcting for the number of genes that we're looking at. And so this is actually really exciting because, you know, we spent 25 years in the wilderness, so this is something to kind of write home about. And so these 5 genes are clearly enriched, but we can kind of take things a little step further. We can try and, you know, take a harder look at, say, the top list of genes, so things with multiple loss of function mutations that are in our sample of 2,200 cases or so. And here we see a much higher rate of, you know, LOFs versus what we would expect. And we can estimate the absolute risk to maybe be somewhere on the order of 20 to 80%. So that is to say, if you have one of these very, very severe putative loss of function mutations, your chance of having autism is not the maybe 1 in 100 or 1 in 88 that we're looking at, but maybe 1 in 5 or maybe 4 in 5. That's a very different sort of kettle of fish. And that's maybe 50 genes or so in that set. And then we can kind of go a little bit further down and look for a little bit more, go a little further down the list of genes that are having these new mutations in the population. And here we see absolute risk on the order of like 3 to 5%. So much smaller chances of having autism, much smaller effect sizes, but there are many, many more genes in this set. So somewhere on the order of, say, 500 or so genes in that set. So again, this is giving us this perspective that the risk to autism in the population is similarly polygenic. It's similarly scattered throughout the genome with lots and lots and lots of small genetic effects. The same sort of story that we were seeing with respect to schizophrenia. This is work that Elise Robinson has been doing in the ATGU with Mark and myself and many others. And this is looking at, you know, intellectual, you know, IQ. So your IQ and intellectual disability, and trying to classify patients into these different, you know, based on how they're going to score on an IQ test. And so if you take patients with so this line up here represents the rate of loss of function mutations seen in intellectual disability. This is the rate that you see overall in autism spectrum disorders from the Simon Simplex collection. And then here's the rate that you would expect for these kinds of mutations. So those 3 lines give you an idea about where you should kind of fall in the scheme of things. And if you take the autism patients with extreme intellectual disability, so measured IQs on the order of, say, 50 or below, you see a rate of loss of function mutations that's very similar to what you're seeing in intellectual disability, giving this idea that there is this spectrum of risk being conferred, perhaps not only to autism, but also to intellectual disabilities for these severe loss of function mutations in the population. But by the time you get to, you know, average or even far above average IQ, IQ of 150 is pretty far above average, you see, you know, the same, if not depletion, of these severe deleterious loss of function mutations in your sample of cases. And so that gives us this idea that there may be other things that are influencing the biology and the nature of these diseases, conditional on what we can learn from the genetics. So we see more severe mutations in autism than expected. We're identifying specific genes. This is very exciting. Maybe 5% of cases have a loss of function mutation that's relevant. Maybe 8% of cases have a missense mutation that's relevant. But this explains a tiny fraction of risk, on the order of a couple of percent. It's not a huge component of risk to disease in the population. And so in conclusion, I'd just like to, again, dwell on this idea that psychiatric genetics is working. We're finding clear regions of the genome that confer risk to these diseases. And that really is a sea change after so long without any really meaningful progress. We're learning new things about biology. There's widespread sharing across multiple different psychiatric illnesses, a point that is, you know, somewhat contrary to the initial articulation of these phenotypes in our classification of mental disease more generally. But there is still a huge amount of work to really translate these findings into understanding about risk, about understanding about biology, about understanding for therapeutics and novel insights to all of these different components that we really need to deliver to improve outcomes for patients and our understanding of what's going on more generally. And with that, I'd just like to acknowledge the kind of PGC, the Autism Sequencing Consortium, and the Simon Simplex, as well as the funding agencies, NIMH and HDRI, and the Simons Foundation. Really, Stefan, Elise, and Caitlin are a dream to work with. It's really fabulous, and so is Mark Daly over at MGH in the Analytic and Translational Genetics Unit. And with that, I'll open for questions. Thank you.
Hi, I have a question, kind of basic question. What is the role of epigenetics in this discussion? In particular, you've identified certain loci that map to behavioral peculiarities. Are these turned on or not turned on? Can you identify them if they're not turned on, and how does that work?
That's great. I think only in the kind of Boston metropolitan area would you get a basic question that starts with epigenetics. It's really good. No, it's an important question. So in terms of, epigenetics is like an entire different and additional field that's really important in the context of how biology works and how cells differentiate and how they decide what functions they're going to do and all of those different sorts of things. In terms of where these genes are operating and when they're operating, that is very much an important kind of subject for research. So with respect to schizophrenia, for example, we took a look at enhancers. Okay, so these are things that kind of help kind of say, hey, I want you to come and make some of this particular gene product. And we looked at cell type specific enhancers across neuronal cells, across immune cells, across a variety of different cells from these large-scale collaborative research enterprises focused on understanding enhancers, things like ENCODE and the epigenomics roadmap. And we took a look at those enhancers, and we saw clear enrichment for the schizophrenia loci in neuronal enhancers, kind of confirming the idea that neuronal cells are probably important in a phenotype like schizophrenia. And we also saw a little bit of signal for the immune system. So where our evidence for immune system and association to the immune system really derives, it derives from this idea that enhancers, that our genome-wide significant loci, seem to be enriched for immune enhancers as well as being enriched for neuronal enhancers. And that's where we get some idea. The expectation is that these genetic variants will either change the gene product, so that would be our missense, reduce the amount, so that's a regulatory variant of some description, be it a loss of function, which really knocks down the amount of gene product you have, or one of these kind of more epigenetic phenomena. And we just don't have the sufficient kind of quality of data or tools at this point in time to conclusively link each of the genetic variants that we've identified as conferring risk to what their specific biological function is. But it's part of, it's along the way, and we're certainly getting an overall view that there is information there to be had.
Man at the mic.
Hello, my name is Ben Bolger, and I was just wondering what type of research is going on right now that looks at genetics and also live brain activity, whether it's MRI, fMRI, or what types of new technologies are being developed that might more effectively assess live cognitive activity?
So let's start with MRI and fMRI. So there are large-scale genome-wide association collaborative activities for structural MRI. So that's measuring how big certain parts of the brain are, how big the brain is overall, doing it in a standardized way to unify the protocols. So there are two consortia working on that. One is called CHARGE, and the other is called Enigma. I think both have their appeals as names. I sometimes wonder if scientists shouldn't be allowed to name things at all, but nevertheless, here we are. That's the world we find ourselves in. And they have identified the first few genome-wide significant loci that clearly influence certain brain volumes as measured in a unified fashion. To work on MRI is like doubly difficult. So genetics isn't cheap. It's getting cheaper, but it's not a cheap activity necessarily. And MRI is like more expensive still. So getting up to scale, and it also takes more time, right? You know, I can just draw your blood and, hi, I got your genotype. That worked pretty well. That's pretty quick. But it's a little harder to say, well, I got to put you in the scanner for, you know, half an hour. Well, it didn't work, maybe a bit longer, and then aggregate that. So it's actually, in a lot of ways, more challenging. But nevertheless, it too is working, and it's working from the same set of tools and approaches that are being applied to other complex traits like schizophrenia and bipolar or diabetes or major depression or what have you. So those things are certainly in the discussion, and what the kind of next round of Enigma in particular is trying to focus on is specific regions of the brain size, right? So you can have your total brain volume, but you also have all of these different parts of the brain that we think do different functions, at least as best we can estimate, typically from things like you have a brain injury and this function breaks, or you know, you have a brain tumor and this function breaks, and that sort of thing. But it's still, I think, a little too early to tell where exactly those MRI and fMRI techniques will fit into our reclassification of risks for disease. But genetics is definitely one of the ways that we can answer that question, and I think that's another reason why having these large-scale, genome-wide significant results is really, really important and valuable, because it does give us a window to see what biology is shared and what's specific and what may be relevant to psychopathology and what might be relevant to other phenotypes that are of interest. So population is a good question. The majority of studies that are contributed to the PGC are European, and that's not actually necessarily by design. It's more a kind of consequence of where available samples with DNA and schizophrenia diagnosis are. And there have been much larger collection efforts over the last 30-odd years with respect to risk to disease, or with respect to identifying genetic risk to disease in European populations than in non-European populations. In terms of different profiles of those risk alleles across different populations, it's a little early to tell, I'd say. It is certainly the case that for other complex traits where there's been a slightly more diverse landscape of populations interrogated at scale, so type 2 diabetes, for example, there are alleles that are very rare in Europe that are more common in Amerindian population, so you know, the kind of that you can pick up through studying large-scale Hispanic activity. So if you go and look at the type 2 diabetes Sigma project, there's a specific variant that is much more common in the Americas than it is in Europe, and that has a effect size that's commensurate with the largest risk effect in a European population. So it's not definitely a guarantee that the allele frequency will be shared across different populations for these risk loci. However, it is the case that where we've looked, for the most part, risk alleles that we observe in Europe are shared in Asian populations and confer similar levels of risk. So that is to say, the effect size seems to be reasonably consistent, as best we can tell, of these common genetic variants that confer risk to disease across the different populations that we look at. So the amount of variability that they might be explaining might change based on how common they are, but they definitely seem to be exerting similar effects across these different environments. Sure.
Thank you. Go ahead.
Hi. So I'm going to ask a somewhat similar question. So the single nucleotide polymorphisms that are putatively associated with an increased risk of schizophrenia, for instance, what sample size are we looking at? What percentage of patients surveyed that have schizophrenia display these polymorphisms? I ask because it would be potentially interesting, I would think, to investigate patients who have multiple of these polymorphisms to see if that leads to potentially a greater risk than you would expect, just simply multiplying the risk of one by the risk of the other, and see if that leads to any sort of commonalities that can give a biological insight.
Sure. So I think there are kind of two questions baked in there. The first is how many what is the distribution of these alleles that we see across cases? And they're very, very similar in frequency in cases compared to controls. They're just slightly but significantly different because we're looking at 38,000 cases and 50-odd thousand controls. There are some cases that have a lot of risk alleles and some cases that don't have as many risk alleles, that have comparatively few in the scheme of things. But we don't necessarily we have yet to identify anything that really kind of would be a secondary clinical characteristic or phenomenon from having more or fewer of these specific alleles in the population. The second part that I think you're asking is something about what we call epistasis as geneticists. So this idea of epistasis has a couple of different meanings, but one of the kind of hallmark meanings is this idea that if I have 3 or 4 specific bad alleles, then I have extremely high risk to disease. So each allele might be very modest, but if we sum them all together, then it's basically a de facto guarantee that you're going to get disease, much greater growth in the risk conferred than you would expect simply from the kind of additive effects or the marginal effects that you get for each allele. To our best evaluation at this point in time, we don't see any kind of scenarios of strong epistasis, but our ability to detect epistasis is actually very small in comparison to our ability to detect what we would call a main effect of the allele. So it is not clear at this point in time that we're seeing clear combinations of alleles that are much more common in some patients versus than others that might isolate a specific biological mechanism or pathway. But it's a hard question to answer because we have very little power. We have very little ability to be able to answer that kind of question itself.
Due presumably to sample size?
Yeah, a lot of it's driven by sample size, but it just is the nature of that kind of test because you're juggling not only one allele, you're now juggling the product of the 2 alleles in terms of frequency. And so if you're looking at 2 10% alleles, you're not looking at 10% of the population or 20% of the population anymore. You're looking at like 4% of the population. Yeah. And so that goes down very, very rapidly. And so that really hurts your power to detect.
Okay, well, thank you.
Sure.
Is it possible to kind of bring all these databases under one umbrella and then kind of correlate with the, if a patient have multiple SNPs, with the like exaggeration of phenotype in them?
Sure. So the great thing about the PGC is that basically all data is shared eventually. So the structure is very federated. Different disease groups, different researchers focused on different disease groups get to define how they want to analyze and process their data and how they want to write their paper and do all that kind of stuff. But they also are very much open, and it's very much kind of written into the ethos and the DNA of the PGC itself, is that the idea that this rolls up to a general, broad evaluation of risk to psychiatric illness. And those efforts are ongoing. Once we establish and identify genetic risk factors, it now becomes a kind of different research question to try and evaluate what specific phenotypes and clinical characteristics and those sorts of things are actually associated with these risk alleles. And so while we have a huge amount of genetic information aggregated and processed and harmonized that has taken 7 or 8 years to kind of get to this point, we're not there with respect to clinical data. So the kind of databases that exist are highly variable, dependent on countries. So there are places like Sweden and Finland where there are medical records that are standardized nationally, where you can start to interrogate these large-scale registers and extract clinical phenotypes that have been collected from patients overall. And then there are places like the US where we're not anywhere close to that sort of thing. So it's a kind of mixed bag with respect to what information we have to pursue the refining of what exactly are these genetic variants doing with respect to an individual.
Where does the trope of the autistic savant fit with our newly enhanced understanding of the disorder?
That's a good question. So the autistic savant, the kind of Rain Man trope, I guess, is probably where it's most got its highest recognition and popularization. It fits in with this idea that what we, while we have limited evidence for what we might term the schizophrenias, I think there's a growing body of evidence that we have what we might term the autisms or the autism spectrum disorders. And so the kind of severe intellectual disability that also presents with repetitive behaviors and complete withdrawal and stereotype movements and things like that, that may exist as one form of autism that we would articulate clinically and evaluate and say and classify. But that doesn't necessarily mean that you can't have retention of cognitive abilities in terms of, or even savantism, where you have a particular skill at memorization or a particular skill at kind of numeracy or something like that, that is necessarily completely divorced from the other. We don't know. Like, it's still very much an open question about how that spectrum evolves over the space of what is causing the biological risk, what is giving rise to the apparent social communication skills, to the kind of inward-looking focus that is consistent with autism. I think that's still very much an open question. I mean, there are growing pieces of evidence that we have a common genetic risk to social communication as a construct. So if you have severely impaired social communication skills, there are common genetic variants that are going to contribute to that in some, not necessarily all, but in some cases. And so we can have that biological process exist without it necessarily interfering with another biological process. Now, it could be the case that there's some amount of comorbidity, right? So there could be the case that what gives rise to your savant ability also has effects on your ability to communicate with others. We don't know enough about the genetics or the biology to really kind of make any sort of assessment or statement about how that fits into the larger landscape.